Hello, this is our lecture number 8 now. We're proceeding along in the area of the genetics of behavior, and this lecture too is one that deals with some basic terminology, and I think probably many of you are already familiar with uh, some of this terminology, but it was important, I think, to make sure that everybody's on the same page. So before we get on into the area of the really interesting twin studies that have been done, what is called concordance research, I wanted to make sure that everybody has the same basic background in terms of some of this terminology. We'll first take a look at a little bit of history here. Certainly, this is a person who's familiar to everyone in terms of the genetics, history of the genetics of behavior. This is Gregor Mendel, who was a monk during the 19th century, and it was his work that really helped us to understand inheritance and how inheritance occurs through genes. If you go back and you take a look at evolutionary theory, especially the theory of Charles Darwin, one of the things that we know is that Darwin didn't really understand how natural selection took place. Indeed, if you take a look at the history prior to the time of Gregor Mendel, it was really thought that inheritance was a product of the mixing or the blending of sperm and eggs. It was really Gregor Mendel who was the first one who really brought us to an understanding, a scientific understanding of what was happening. So when you take a look at his work, his work tells us that genes are really the basic units of heredity, that their structural identity doesn't really change at all from one generation to another. We have, many genes that are aligned along chromosomes. Chromosomes consist of these strands of thousands of genes, and they come in pairs. And it was Gregor Mendel's work that was done principally through plants and understanding the size of seeds and their color, and he was able to determine how this inheritance actually occurred. And again, this work initially was done with plants. So when you take a look at, some of this important terminology, certainly, when we talk about genes, we're talking about them being a portion of chromosomes. Chromosomes consist of what we call DNA, deoxyribonucleic acid. And DNA is the model for the synthesis of ribonucleic acid. So when we take a look then, this is our DNA. This is a self-replicating molecule. And each base determines one base of RNA. So we're getting a copy now of one strand of DNA, and it's through these proteins then that become a part of the body's structure. Others are enzymes that control the rate of chemical reactions. So again, when we take a look at what's happening here in this duplication process that occurs, this copying process that occurs, this of course is all being driven by these self-replicating molecules that we call DNA. When you take a look further at this process, RNA is serving as this very important template. It's serving as a model. And that is an important process in terms of the synthesis of proteins. When we explore the contribution of these proteins to the development of the body, They're involved in the formation of the structure of the body. They're involved in terms of serving as catalysts, biological catalysts that are regulating chemical reactions in the body. So again, this template then, this RNA is serving as this template for the synthesis of new proteins. And those proteins are really doing a lot in terms of determining the structure of the central nervous system, in terms of regulating certain reactions, chemical reactions within the body. So this discovery then was, a crucial one in terms of helping us to understand ultimately the genetics of behavior. There's again some terminology here that's important. One is this whole idea of homozygous genes and heterozygous genes. Homozygous genes means that a person has an identical pair of genes on 2 chromosomes. Heterozygous for a gene means that a person has an unmatched pair of genes on the 2 chromosomes. So you can see the diagram here on the body, here on the bottom of this slide. Here we have the homozygous situation here where we have identical pairs of genes on the 2 chromosomes. We have the same thing here in this condition. And here we have the heterozygous condition where you have two different, you have unmatched pair of genes on the 2 chromosomes. So again, we can have genes that are either homozygous or heterozygous. Genes can be either dominant or recessive. And when you take a look at the numerous, examples of it, and that's seen on the upper right-hand portion of the slide. Here, this is a dominant gene effect that you see right here, the cleft in the chin. This is recessive in the case where an individual does not have this. Widow's peak that you see here. No widow's peak that you see here. Again, this is recessive, this is dominant. Dimples, that's also dominant. No dimples, that's a recessive condition. Freckles, eye color, whether the earlobe is free, as it is here, or whether it's attached. Again, these are all related to dominant or recessive genes. So some genes can be either dominant or recessive. And again, the examples of that are what we see here. A dominant gene shows a very strong effect in either the homozygous or the heterozygous condition. A recessive gene is really only showing its effect in the homozygous condition. So again, understanding, some of the basics there in terms of dominant recessive genes, I think is important to understand as we move along. When we take a look at the outcome of mating of parents who are heterozygous for a particular gene, again, it was Gregor Mendel who really helped us to understand this. But just to give you a little bit of an example of how this works in terms of determining what happens in terms of offspring. There's been a lot of interesting work that's been done with the ability to taste and the ability to taste certain substances that are bitter, like PTC, for example. And here is the PTC molecule that you see right here. We have a gene that helps us in terms of our ability to taste substances like this that are bitter. And those bitter tasting substances are in a lot of different things, like coffee, for example, like certain vegetables, like Brussels sprouts. This is a gene, this tasting gene, which is represented in the population And we can be either heterozygous for that particular gene, or we can be homozygous for that particular gene. So if we take a look at the work that has been done in this area where the father and the mother are both heterozygous tasters, that is, they both express this gene in terms of the ability to taste this bitter tasting substances. If you were to take a look at their offspring, in terms of how it is represented in their offspring, you would see something that would look like this. You would have homozygous tasters that would represent about 25%, where you have the dominant gene and a 25% chance of being homozygous for the recessive gene that you see right here. So that consists of 50% probability. And you would see that the heterozygous condition that you see right here, where an individual has the dominant and recessive, you would have, again, a 50% likelihood that that individual, that you would have those individuals being represented in the offspring probabilities. So all in all, then, you would have this 75% of the offspring would be able to taste this substance. Non-tasters, again, would be this recessive condition that you see here, both recessive genes, so 25% would not be able to taste. So again, this is, looking at probabilities that occur in terms of the outcome of mating between parents who are heterozygous for that given gene. So it was Gregor Mendel, again, that kind of brought us to this point in terms of our understanding of dominant and recessive genes. Another important phenomenon here is the phenomenon of what's called crossing over. And this is something that occurs during meiosis. Each chromosome participates in reproduction independently of others. So during meiosis, then you have pairs of chromosomes. They line up with each other. They duplicate or replicate, as you see here. And then one chromosome crosses over the other, as you see here. And during that crossing over process, they break apart, and the genetic material is shuffled. And again, that is why no offspring are perfect matches to their parents, because of this phenomena of crossing over that occurs, and the outcome in terms of the shuffling of genetic material. So again, each species has a certain number of chromosomes. In the case of the human being, our cells, each one has 23 pair. And this crossing over occurs during meiosis, and it's important for the shuffling that occurs of genetic material. We should also explore these two other important concepts of what we call autosomal and sex-linked genes. And that is represented in this diagram that you see right here. So when we take a look at different types of genes, some are autosomal genes. That's all the genes really except for sex-linked genes. And again, that's the majority of traits that come from parents. And we also have sex-linked genes. These are genes that are located along the sex chromosomes, and they're really highly dependent upon the sex of the parent. So in the case of mammals, human beings are mammals. Sex chromosomes are designated as being X or Y. And females have 2 X chromosomes, and it's males that have X and Y chromosomes. So it's really the male then that's going to be determining the sex of the offspring. So when we explore this determination that you see here, and I think that it's represented very well in this diagram here. Here we have a female contributing, being designated as XX, male being designated as XY. And females are contributing, these X chromosomes. Males can contribute either an X or a Y. And again, it's the male that's really going to be determining the sex of the offspring. So we have a 50% likelihood of female and a 50% likelihood of male offspring. So again, if an X chromosome is contributed by the male, the offspring is female. If a Y chromosome is contributed by the male, the offspring will be male. So again, just a little bit of the basics there in terms of our understanding of determining the sex of offspring. When we talk about the formation of proteins, and again, this whole process of development is crucially dependent upon those chromosomes and the encoding that takes place in terms of proteins. On the Y chromosome, there's at least 27 proteins. The X chromosome has genes for approximately 1,500 proteins. Again, this is very important to understand this because it is these proteins that are going to be so important in terms of neurodevelopment. So sex-linked genes usually refer to X-linked genes. For example, things like red-green color deficiency, that's a sex-linked genetic conference that occurs. And sex-limited genes are genes that are present in both sexes, but they usually just have an effect on one sex. And again, to give you some examples, things like chest hair, breast size. These are sex-limited genes. Sources of genetic variation, that's certainly a very important topic here that we're going to pick up on in our next lecture after this, to try to understand this a little bit better. But this is really what allows evolution to occur, are these different sources of variation. And again, when we take a look at recombination, and what we're doing here is taking a look at this crossing over process that occurs during meiosis. This obviously is a huge source of genetic variation. So again, you have new combinations of genes in offspring. And this is why offspring are not identical to that of their parents. And it's because of the shuffling that occurs during meiosis. And there's also mutations. These are quite rare. They're random. Oftentimes they run against what the organism actually needs. They are, again, random events, very rare events, but this is another source of variation. So in our next lecture, we're going to be exploring some of the wonderful twin studies, concordance research that has been done. And again, there's a number of videos that you will watch in this area that I think that are important as background. You should watch those prior to the time that you watch the next lecture. Again, in the syllabus, there's these thought questions as well as the videos that I've archived that you should also watch in addition to the reading that you're doing. So this is the end of this particular lecture, and we'll take a look at twin studies, the concordance studies, identical twins or fraternal twins. That'll be one of the important topics in our next lecture.